Hi, I'm Steve Mirsky. How you doing out there, everybody? And welcome to another edition of 18 Minutes With, co-sponsored by the Kavli Prize. And I'm with co-sponsor Scientific American Custom Media. The Kavli Prize recognizes scientists for pioneering advances in astrophysics, nanoscience, and neuroscience. It's a partnership among the Norwegian Academy of Science and Letters, the Norwegian Ministry of Education and Research, and the US-based Kavli Foundation. For today's chat, we welcome Corey Bargmann, a co-recipient of the 2012 Kavli Prize in Neuroscience for revealing basics about how a nervous system works to create perception and allow decision-making, in other words, behavior. She's head of science for the Chan Zuckerberg Institute Initiative out of her lab at Rockefeller University in New York City. Hi, Dr. Bargmann. How are you today?
I'm very good. Hi, Steve. How are you?
Very good, thank you. It's really good to see you. Your first job was making fly food in a laboratory for, I assume, Drosophila. And then you moved on to an even simpler organism, C. elegans, a roundworm. Tell us about C. elegans and why it has become such an important model organism for scientists to work with.
Well, if you look at a picture of C. elegans, which we should have available here, you can see that this beautiful creature was named elegans for a reason. It's a graceful organism. I think we have a slide that we can show, an image. And one of the things that's really striking about this animal here, as you look at it with its nose to the left and its tail to the right, and it's throwing off a couple little offspring there, is that this animal is completely transparent. You're just looking at it through a microscope and you can see every organ in its body. If you look at higher magnification, the sort of fuzzy stuff near the nose is the brain. You're looking at the gonads and the gut running through the whole body. It's a gift to biologists to have this kind of ability to look inside the animal's body, to watch biological processes without disturbing them. In fact, with modern technology, we can even monitor the activity of the brain of this animal. We can literally watch what it's doing while it's thinking. And that provides just the ability to move back and forth between different biological questions and the whole animal in a way that's very exciting. And it's especially important when you're trying to think of very complex questions like behavior, which you always have to break down in some way. You can't study all the behaviors and all the neurons and all of the neural activity in a human being at the same time, but you can study all of those realistically in a worm at the same time. And you can really try to move across levels to see how these things are put together.
And crucially, they have many of the same molecules that we have and that all animals have. So how does that work? Why are those molecules maintained over evolutionary distances? They must be really important.
Yes, I think one of the biggest surprises of the Genome Project about 20 years ago, that came as a surprise even to those of us who worked on worms and flies, is how much of the basic machinery of genetics, of genes, was already present in very simple animals and is shared between almost all animals and ourselves. And so if there are, say, 25,000 genes in a human being, there are also 25,000 genes in a worm. And at least 2/3 or 3/4 of them, you can recognize them across that huge distance and see that a particular gene for a particular calcium channel is recognizably the same version of a calcium channel that would be present in a human and in a worm. So this is shocking because we are quite different animals. And one way to think about that is that the genes represent something like the vocabulary of biology. They represent a series of concepts that you can string together to generate different kinds of functions. And you can use the same vocabulary to write the works of Shakespeare that you can use to write Dr. Seuss. And the words mean the same thing in these different languages. And so you can try to translate between what a gene means in a simple animal like a worm, what it means in an intermediate animal like a fish, and what it means in a very complex organism like ourselves, to try to translate those different pieces of information.
Yeah, it's really quite amazing. But there are differences, and you have said that you don't believe, for example, that C. elegans experiences, for example, depression. Now, they might be using the same molecules or very similar molecules, but to somewhat different effects within their more limited physical structure.
Well, here's a way of thinking about that. There are certain molecules that affect behavior across many, many different animals. And you would think, well, how similar are they? How similar are the functions? And I want to talk about one of them. I have a slide illustrating that. And that's a very famous molecule in the nervous system. Even many non-scientists have heard of it, and it's called oxytocin. So I have a slide that sort of illustrates something about oxytocin. So oxytocin is a small molecule that is released by the brain when a female mammal gives birth. And it helps the uterus contract, it helps the mother nurse her young. It also modifies her behavior so that she has very complex social behaviors associated with rearing her offspring. And on the left side here, you actually see a little family of voles, small rodents, where the mother and the father raise the offspring together. And oxytocin is part of the more complex social structure that they use as well. So oxytocin is also present, it turns out, in a worm. Pretty surprising. And here on the right here, you see the equivalent of what a worm family might look like, which is the female-like hermaphrodite, and accompanying her is a male. And the mating behavior that links the male to this hermaphrodite, to this female, and this mating pattern is regulated by oxytocin. So social behavior in voles, social reproductive offspring in people, and mating behavior in worms is linked together by the use of a molecule that functions in the nervous system to tell the animal that it's in a social context and thereby modifies its behavior. The social context of a worm is nothing at all like the social context of a rodent, let alone or of a honeybee or of a human being but you sort of start to see that certain molecules got locked into that very early in evolution. And once they were assigned to that very important biological activity, because social behavior is incredibly important to all species, including ourselves, you couldn't very easily remove them without causing some disruption. And so you just kept reusing them over and over, building more and more complex structures on top of them. And similar sorts of ideas emerge for other kinds of functions as well. I think oxytocin may be the most compelling sort of single relationship of a molecule to a particular function. But there are some others as well, that certain kinds of signals are propagated in similar ways.
So how does studying the systems that have analogs in other animals in this tiny little worm, how does that inform what we know about the more complex creatures that are out there, like the mice, the voles, the people?
So here is where we think about problems differently based on the tools that we have available in different organisms. And in C. elegans in particular, for over 30 years, there has been a tool available called the connectome. And what the connectome is a complete map of the connections between every single nerve cell in the worm's brain. And so these diagrams here are actually diagrams of every neuron in a worm. Each one of them is illustrated by this small, like, illegible group of numbers, like right in the center up at the top there, I think with ASJ left. And there are two things to say about this. First of all, there are 302 neurons in a worm. There are 86 billion neurons in a human being. In a worm, we can really think about what every single neuron is doing at the same time. That's just not possible in a brain as complex as our own. Second thing, we know when we are studying the neuron called ASJL that we are studying exactly the same neuron in every worm. That if I go back to that same neuron tomorrow or the day after that in a different animal, it will be the same. And so I can develop a body of knowledge about every neuron. Again, 86 billion neurons, it's much harder to even find one neuron reliably over time. So we can develop, and we can also know that if I'm studying this neuron, and you are studying this neuron, our information can be put together. So we can increase our knowledge that way by working together. And then the third thing is that we know the connections that every single one of those neurons makes with other neurons. So we literally know all contacts that they make with other neurons, where those contacts are, how strong they are. And those are reliable, although not perfectly identical, from animal to animal. And so we can follow the flow of information through the brain. And that is an incredibly challenging, fascinating, difficult problem. And
I should say that folks watching can put questions forward. I believe there should be some instruction on your screen, and you can ask a question that'll possibly get squeezed in here. One question did come through already, and that is, you mentioned oxytocin, and somebody wanted to know, isn't that in humans one of the key compounds in bonding between a mother and a baby?
yes, that is, we do believe that oxytocin plays an important biological role as one of the underpinnings of the complex human behavior of bonding. So I don't think it's, and this is what I said, I don't think worms get depressed. I don't think worms express bonding in the same way that we do. But there's a basic biology that tends to bring together and align the behaviors of individuals whose behaviors should be aligned to one another. So in our case, we develop complex family structures and lifelong relationships through this bonding process. And somewhere deep at the root of that, there's a little piece of biology that helps to build that relationship and strengthen it over time. And that is what the oxytocin is doing. And that little building block, that little basic piece of vocabulary, you would find in a vole, you would find in a sheep. Oxytocin was first discovered in sheep. Yeah. And you will find in a worm.
Your Kavli Prize was for what exactly in this world of research?
My Kavli Prize was for really using the wiring diagram and the genes of the worm to understand, starting from how a behavior is assembled and becomes more complex over time. So an example of the kinds of things we study and the way that that relates to molecules like oxytocin is in this slide here. So we can study the behavior of worms, for example, looking at a particular chemical. And we can say that animals release pheromones, and those pheromones are chemicals, and those chemicals are detected by a particular neuron. In this case, I can tell you in every worm that a particular neuron will sense particular pheromones. Then we can ask questions about how those neurons are related to behavior. So we did a series of experiments that showed that neurons are hardwired into certain behavioral responses. So for example, we found that certain food odors were always attractive and other odors were always repulsive. And we determined that the logic of that started right out at the very first sensory neuron that detected the odor. That the sensory neuron was devoted to either attraction toward, acceptance of, or rejection of, and avoidance of a substance. And that was hardwired into the nervous system. And that turns out to be a general principle in the olfactory and taste system. It was subsequently shown that, for example, sweet taste and bitter taste are just hardwired into the brain. But then we know that behaviors are flexible. We know that behaviors can change. We know that not every time a person or an animal sees a particular stimulus does it respond the same way. And so we asked how it was that those changes could take place. And that led us back to molecules like oxytocin. So what we found is that certain neurons contained within their wiring, the complete wiring for two different behaviors. So pheromones, for example, can be either very attractive or they can be quite repulsive. And that's present in the wiring. You can see that wiring coming right out of that first olfactory neuron that could go either toward neurons involved in attraction or neurons involved in escape. And then you ask yourself, well, how does that work? And it turns out that what the neuropeptides do, what molecules like oxytocin do, is to choose between those two outcomes. is that when they are present and convey certain kinds of information, one of those systems is active and the other is not. So in the particular case of pheromones, we can have animals, in this case, avoiding the pheromones, in which case they'll be solitary and they'll just scatter very broadly. But under other circumstances, they will aggregate. They will be attracted to the same pheromones. It's the same olfactory neuron. It's choosing between two different classes of its possible targets. And the choice between those targets is based on another factor that comes in. And that factor, in this case, has to do with stress response. Stress response is a very strong stimulator of aggregation behavior. And that stress runs through the regulation of a modulatory molecule, a neuromodulator. So a molecule in that same family as oxytocin, to modify and reshape the functions of the nervous system. So every worm has the potential to do both, and the neuropeptides to sort of select among those connections to allow different behaviors to happen at different times. And if you'd like to see what worms look like when they're excited about other worms and their pheromones, we have a little movie of worms doing exactly that, with two of them colored in white with the transgene to make them easy to see. And what you can see is that these guys really like hanging out with each other. And they gather together, they're sensing pheromones. Every now and then somebody goes off to check his voicemail or something, but then they come back, they join the group. And this kind of aggregation behavior is regulated. It's regulated by the environment, like stress causes this to be stimulated. It's also regulated by the genetic differences between individuals. So we study largely in the lab, we study a strain from the UK, from England. It's, these guys are kind of solitary, they're loners, they're a bit standoffish. But we can also study a strain that comes from Hawaii that we studied quite a bit. And those guys aggregate all the time, very sociable worms. So
that's pretty interesting. You much earlier you mentioned them thinking, which I don't think you mean in the same sense as when we think, obviously. But so what do we mean by them thinking, making decisions based on their environment?
Yes, you can start with the idea of taking information from the environment and making decisions. But one of the really fascinating things that's happened in neuroscience as a field, and that is one of the adventures for the future in this field, is the fact that we've realized that the old formulation that it's like sensory information comes in and behavior goes out is not probably the right way to think about most behaviors. Most behaviors come from ongoing activity in the nervous system, self-generated activity, patterns of activity, and then sensory inputs modify those. And that process of having some kind of unified information flowing through the brain actively at all times is just one of the mysteries of neuroscience. People refer to it in different ways. In human fMRI, you might refer to it as resting state activity, where you see large-scale activity patterns in the brain. In certain theoretical constructs, it's called manifolds or low-dimensional representations. But they all have to do with the fact that activity is flowing through the nervous system constantly, and that new information can come in and modify that. But the overall active pattern, whatever it is that's going on in many neurons simultaneously, is just a fascinating direction for future understanding.
Right, and it can't be locked in, or else the organism will be inflexible. And you need that flexibility for survival, I would assume.
That's right. Every animal gets very good at solving the problems that it encounters in its own environment. That's what evolution helps it to do.
Right,
and if the environment is not constant, then the animal can't always do the same thing either.
You're a long way from retirement, but what do you still hope to accomplish in your remaining years in the lab?
I would really love to make some more insight into understanding how it is that the neuromodulator logic, the logic of these molecules, of neuropeptides and other molecules like serotonin, connects with the fixed wiring diagram. I think that they're special neurons, they have separate properties, and we still really need to understand how these systems are modifying what the nervous system is doing. And I think that's a fascinating question to ask. I think about it particularly in the relationship we have between the brain and the body, that these kinds of signals are often carried through these sorts of modulatory systems that are sort of linking together physiological processes and mental processes. And I think making some progress on that might give us some insights into some of these questions like depression, that are really, clearly related to the functions of the body and not just of the mind.
Sure. I would assume that your kind of work is of great interest to people studying addiction or eating disorders. I mean, basically anything that involves human behavior, but especially things like that, where any kind of intercession might be really helpful to a lot of people.
I have had the experience in my own career of having worked on a basic science problem. It was when I was a graduate student working on cancer that ultimately led to clinical advances that are used to treat human beings today. Identifying a gene called Herceptin that is at the basis, a gene called HER2 that is the basis of a breast cancer treatment called Herceptin. And so I played a small part in that. And so I really, really believe that basic science leads to advances in human health in the long term. But I have to say, I also believe that when you study the brain, and I believe that studying the brain will ultimately give us a lot of insights into brain disorders, which is very important and which is a huge source of suffering for many people, whether it's mental health or neurodegeneration or autism, developmental disorders. But at the same time, I really believe that studying the brain is fascinating and beautiful and mysterious and something we should all want to do. And, what could be more important than knowing how the brain works? What could be more fascinating? What could be better than knowing how your husband's brain works or how your teenager's brain works?
That would be cheating. I was going to say, we all have one, so it's good to have an owner's manual. I just want to, well, thank you very much. This has been great fun. One other thing, nominations for the next Kavli Prizes are open. What has the prize meant for you, and why would you hope that others might take the time to nominate researchers whose work they admire?
The Kavli Prize does take this attitude that science is beautiful and exciting and engaging and awesome. It's astrophysics, it's studying the planets, or it's nanotechnology, all of these incredible little machines you could build, or it's neuroscience, studying the most complex system we know about, which is the human brain. And people should nominate for the Kavli Prize because you want to show attention and appreciation and excitement about these fields. You want to bring those to other people. You want to show people that these advances are human accomplishments that we can celebrate. I can tell you one little story, if we still have time, about winning the Kavli Prize.
Stretch it a little bit, sure.
I met Millie Dresselhaus won the Kavli Prize in nanotechnology the same year that I won it. And she was known as the Queen of Carbon.
Yes.
She was an MIT scientist, an extraordinary person who worked on nanotubes and graphene and things like that. And we met the King and Queen of Norway, and there was a big reception. Everyone was all dressed up, and we were all introduced. And King Harald of Norway saw Millie Dresselhaus, and he said, "I believe you are the Queen of Carbon." And she said, "Yes, they call me that." And he said, "King of Norway," and he bowed.
It's a great story. That's great. I believe there's a biography of Dresselhaus about to come out, if it hasn't already come out, any day now. So look for that. I have to apologize. I missed a couple of notes that people in the audience were asking a question. Can we go on for another couple of minutes? Folks in the truck, as they say. Well, there's not really a truck. The brain, the human brain, we're not going to be able to get a connectome for the human brain anytime soon, I would assume.
We're working on it. We got the worm brain in 1986. Then we got the fly brain a year or two ago. We're just putting that together now. That's 200,000 neurons instead of 300. Still got like a long way to go between 200,000 and 86 billion. But you figure out more how to advance it. I'm not sure. I don't think I'll see a human brain within 10 years, but science is somehow always surprise me with what you can do. And let's say that maybe in 20 we could start to see what that looks like, at least overall.
That would be brain-boggling, in fact. When you look at the same neuron, I'm not sure this is the right way to say it, but in different animals have their own sets of neurons. The synapse assortment, that has to be very different too, right? And that makes it, I mean, you were just talking about the connectome. So what we're basically talking about is a connectome in different animals is very different because just the sheer number of neurons in different animals is quite different.
Yes, the connectome is different in different animals, but one of the things that you see that allows the brain to evolve to the complexity it sees now is that it's modular. So you'll see a little module of neurons connected in a particular way that's repeated over and over and over again. So for example, there are maybe 10 million neurons in the human retina, but they actually consist of, a motif of maybe 100, 200, 500 neurons repeated over and over again in little blocks, like a checkerboard.
Mm-hmm.
So that was why I said we might be moving toward this. We don't have to look at every single connection in the eye. Maybe if we just solve a couple of those little, 500,000 neuron motifs, we'll be able to kind of more or less estimate what the rest look like. So many, many repetitions, lots of parallel processing, as we say in the computer field, is one of the things that allows the brain to evolve to greater size without having to reinvent everything every time it makes a few new neurons.
And one other thing, it's a slight variation on something we already talked about, but can oxytocin levels be affected strictly by the environment?
Yes, there are fascinating studies in rats that show, so first of all, of course, when a female rat gives birth, her oxytocin levels surge, as they do in every female mammal, and she bonds with the pups. But virgin rats, younger females, they're not very interested in pups. But if a virgin rat hangs out with a mother and her pups for a while, she gets kind of enlisted in the rearing process, and her oxytocin levels start to rise.
It's great.
It's great.
So fascinating. Dr. Bargmann, thank you so much for your time and obviously your expertise today. It was great talking to you, and I hope everybody who was in our audience comes away with a feeling of bonding toward their fellow human beings and this amazing network of neurons that we all share.
Thank you. Wonderful conversation. Thank you as well.
Take care. Take care, everybody. We'll see you next month.